Very good morning all. Today I welcome all national and international participants for this workshop. So today's first lecture, introductory lecture will be given by Dr. Siris Srivastav. So here I am going to give his brief introduction. Dr. Siris Srivastav had earned his Master in Chemical Engineering from Indian Institute of Science, Bangalore, where even though new to chemical engineering, he published 3 research articles. He earned his PhD in Chemical Engineering from Michigan State University. At MSU, he won the Sigma Chi Award for Excellence in Graduate Studies. He conducted his postdoctoral research work in the National Institute of Health in the area of metabolism and won the Fellows Award for Research Excellence for his research in 2011. He joined ICGV New Delhi as a team leader in 2012. At present, he is a group leader of Systems Biology for Biofuels group at ICGV New Delhi. Research in his group relates to improving growth or product formation rates through application of system biology and metabolic engineering. His group has created and analyzed genome scale metabolic model of variety of biotechnologically important organisms. Other work in the group involves 13C metabolic flux analysis of both heterotrophs and photoautotrophs as well as stress adaptation of organisms to increase growth and product formation rates under industrially relevant conditions. He has published over 35 research articles in reputed peer-reviewed international journals and 1 patent application. He is a review editor in Systems Microbiology in Frontiers Group of Journal and a member of several institutional committees. Thank you. I would like to invite Dr. Siris Srivastav.
Thank you for the introduction. And well, first of all, I must formally welcome each one of you here, and both who are physically present and also several of you who have joined through our Zoom link, as well as those who are utilizing our YouTube link to see this meeting and conference. So first of all, I must say that this has been a little bit long journey to get you guys here, but we are absolutely glad that all of you are here, both who are physically present as well as those who have joined us through Zoom. I know some of you must have applied for about over a year earlier, and I'm very happy for your continued interest in this workshop, and we'll try our best to make sure that everyone gets a fair output from this workshop. Of course, for events like these, your feedback is also very, very important for us to improve in future, what we can do better, what aspects we need to improve, what are the areas where we need to focus. But I guess I think it's very, very useful that we have this workshop in the upcoming area of bioenergy. Of course, this workshop is possible because of the joint efforts by both ICGB and Department of Biotechnology. The Department of Biotechnology of India has funded this workshop, and ICGB has conducted a series of workshops in which this is the first one. So, and in terms of organization, since this is a course, we have organized the course primarily in the morning as well as afternoon sessions. So morning sessions typically will have 2 lectures each day, and we have, and if you guys have seen the booklet or the workshop program, you must see that there is a very exciting group of lectures which will be there from a lot of reputed scientists, both in the area of systems biology as well as synthetic biology. Now these so-called theoretical lectures or talks which tell us about the various applications of these modern methods and techniques will be supplemented by hands-on training, which will be done in the afternoon session. So our goal is to give each one of you a fair sense of what we go through on a regular day, what are our exciting approaches as well as our challenges and limitations. So one of the primary goals of this workshop also is to give you guys a platform for all of you who have similar interest, who shares interest in the similar area, to also network and collaborate for future work. So definitely the strong recommendation would be for each one of us to utilize this opportunity to maximum, both in terms of as much discussion as we can do, so as to gain output both for ourselves but also share whatever we know. So with that, since our workshop is, or the course is titled Flux Applications of Flux Analysis and Synthetic Biology in Bioenergy, so I think based on this I thought that I must first discuss certain fundamentals of flux analysis and its applications in bioenergy research. Now obviously you will be hearing a lot of talks from many experts in this field who will be talking much more in detail about various applications of these techniques, methods in bioenergy as well as biochemical production. So that will give you a perspective on what we can do with these. Now before that, it's possible that not all of you have background or significant training in flux analysis. So based on that, I have decided to cover some certain fundamentals of flux analysis and also what kind of work we have done in terms of their applications in bioenergy research. So today I'll be talking primarily about genome scale metabolic models and flux balance analysis, and how what we have done has given us some very useful insights into the biochemical processes as well as helping to design the metabolic engineering strategies. So obviously bioenergy throughout the world is a very important energy resource, and just to cover the basics of bioenergy, it's defined as renewable energy that is derived from recently living organic materials known as biomass. And according to rough calculations, a ton of biomass could produce about 50 gallons of biofuels, more than about 50 pounds of various bio-based chemicals and bioproducts, or if simply burned efficiently, it can generate about 85 kilowatt hours of energy. Now, if we talk specifically about India, now obviously as I told that a lot of developing countries utilize biomass as their primary energy resource, mostly through direct burning. Now in this case, obviously there are issues in that burning is not efficient, so it produces a lot of pollution. And so the idea is, can we, instead of polluting or utilizing it effectively, it's burning the biomass inefficiently, can we convert it into various kinds of fuels to help reduce the energy environment as well as protect the environment from the excessive CO2 emissions that are due to use of fossil fuels. So if we take just in case of India, so India, the crop residue biomass on an annual basis is about 700 metric tons, out of which about a third is surplus, which can in potential be used for bioenergy generation. And according to International Food Policy Research Institute, about 30 billion annual loss is caused due to stubble burning. Now it's the same thing, which is a problem but also can be a very useful resource, that if we can probably utilize it efficiently, we can very likely supplement about 17% of total primary energy consumption of the country. Now these figures are more for India to give a perspective, but for any economy or any country where agriculture is a primary mode of earning, these kind of figures are fairly comparable. So this approach or this general concept is very much valid throughout the world. So obviously, since biofuels are considered as important important resources for reducing the pollution as well as utilizing the biomass. I just will briefly cover what are the various biofuel generations and their challenges. So of course you have the 1G ethanol, or which are primarily based from sugar-rich substrates such as molasses, corn, starch, waste grains, etc. And these are in that sense, these are simple fermentations from our biotechnology perspective. However, the major challenge here is that, especially for yeast, which is the primary organism for fermentation, the performance of the yeast drops under high concentration of sugar. So if you typically, once you do fermentation of about 10% sugars, everything is fine, yeast is very happy and you can get the fermentation very fast. However, for improving the economics of the process, you would want to use the smaller reactors, but then if you want to use a smaller reactor, you need to increase the concentration of the sugar. And once you do that, so for example, if you ferment a 30% sugar solution, obviously, so 30% sugar solution will give you about 15% of ethanol, which is what you typically get in your wine. Now, under those conditions, the fermentation performance of many yeast significantly reduced compared to what they do with 10%. So, also 1G ethanol is actually the first step towards ethanol independence, where even in India, a lot of 1G ethanol plants are being put up by a lot of sugar mills as a supplement to their income. So obviously it's not that, even though it's a very simple technology, it still has a lot of relevance in terms of biofuel production. A main interest of recent years is 2G ethanol, or which is primarily based on agricultural residues. Here again, the fermentation is by yeast, but mostly by engineered yeast, so that they can utilize the xylose that is present in the mixture of hydrolysate of biomass residue. And here, enzyme for biomass hydrolysis is a major contributor of cost. But of course, you also need to design your yeast so that it can efficiently use xylose as well, because traditionally or naturally the yeast does not have the set of enzyme. When I talk about yeast, I am talking about Saccharomyces cerevisiae. It does not have the set of enzymes which is required for xylose fermentation. Obviously, since effectively all this kind of fermentation is utilization of sun energy in one form or the another, so there has also been effort in terms of utilizing photosynthetic organisms for production of different kinds of biofuels, in which case, for example, algae with this raceway ponds and all are getting traction in terms of research, though of course given the prohibitive cost and low concentration of biomass, until now algal based biofuels have not been very successful. But effectively the idea here is to generate triacyl glycerol or tags in the biomass, which can then be used with extraction and transesterification to produce biodiesel. Of course, there are several challenges here which involve very slow growth as well as harvesting a biomass which is primarily single cell organism. The further advanced processes include engineered algae and cyanobacteria for direct photosynthetic production of biofuel. Until now, again, there are several proof of concepts in this area where, for example, algae or cyanobacteria have been engineered to increase their production of the molecules of interest, and also for heterologous production of molecules. However, obviously the rates and titers are still too low for them to be really viable, but these two are also intense areas of research in the area of bioenergy. So obviously a typical, is there a question or something? Yeah, so I'll answer the question later. So the question is, will it be available on YouTube later? Yes, definitely it will be available on YouTube later. completely because the experimental sections required to be done in labs, so that part will not be available on YouTube immediately. We aim to generate some videos and then post them online later. So of course your typical process requires chemical pretreatment and enzymatic hydrolysis to release this C5 and C6 sugars as well as lignin monomers. Lignin is separated and the sugars are fermented. Now the idea here is that, okay, once we have the sugars, we are not restricted to biofuels if we have information of the metabolic network of the organism. So we can produce biofuels, biochemicals as well as biodegradable plastics. So again, a lot of our traditional unsustainable processes can in fact be altered to make them sustainable. Of course, the challenge here is to make them cost effectively. On the other hand, primarily with the photosynthetic organism, one can then make sugars, lipids and proteins as well as direct photosynthetic production of biodegradable plastics and the chemicals as well as biofertilizers. So, so in terms of the whole idea of the recent efforts in bioenergy, metabolic engineering as applied to bioenergy is, can we not burn the stubble that we have and instead utilize it to get the sugars and then utilize the plethora of information that is available about the biochemical pathways to make the products of interest, whether biochemicals, biofuels or biodegradable plastics. But of course this is easier said than done because it will require a lot of metabolic engineering and synthetic biology efforts to produce the compounds of interest in a cost effective manner. So now whenever we are talking about any bioprocess, irrespective of whichever process we are talking about, it has to be right. And by right, I mean it should have a perfect balance of rates, yields and titers. So of course the rate of a bioprocess determines how fast we can make the product, which effectively determines what are the volumes or cycles that we can make per year. So for processes or molecules such as biofuel molecules, you need to understand that we are talking about millions and millions of tons of product per year. So your rate has to be very fast. Of course, the biggest challenge with any of the biofuel molecules is you are competing against something that is practically drilled from the ground and then refined and then sold. So versus there are so many steps in bioenergy processes. So rate will determine how much volume I can make per year and obviously how fast we can make. The yield obviously will tell you how much we can make per gram of the, generally it is defined in terms of the costliest component. So obviously this will, higher the yield, the better the cost effectiveness as well as the purity. So for downstream purposes also you want to improve yield. So if for example a typical fermentation with E. coli is a mixed acid fermentation which produces a variety of compound, if we can perhaps engineer the organism to make or most effectively make the product of interest, then the purity also improves, which improves its downstream effectiveness. And of course the titer. So what is the concentration of products which we can achieve? Again, this has a implication in downstream effectiveness as well as the volume of the, so ideally we want the titer of the product to be as high as possible, and I gave you an example of ethanol as the case where you can ferment 10% ethanol but you really want to ferment 30 or 40% for glucose or sugars. So any biological processes need to go through a cycle of what we call as DBTL, which is design, build, test and learn. So here in this course we will be going primarily through the first three sections of this. Obviously learning means once you do all of these, then you check the performance of the organism or the process that you have made and then further or modify whatever is needed. So in terms of design, of course, the first thing is selecting optimal host, which has certain basic characteristics in terms of product formation or robustness to certain bioprocesses, as well as resistance to the, say, many times the product itself is inhibitory to growth. So all those characteristics are required in the host, and of course then you kind of design your set of engineering where you want to motors I want to use, what RBS I want to use, and so on. Here in this work, in this thing, we will be course will be talking a lot about various modeling approaches. So again, what are the biological pathways that are present and how do we manipulate them in order to make the product of interest. So a little bit also about pathway selection we'll be talking, and not only you will be doing some of these exercises in the afternoon sessions, and then of course in the synthetic biology section we'll be talking and doing a little bit about parts assembly and host modification. How do we engineer using CRISPR-Cas based system an organism to produce what we want, and of course testing. So whatever we test, we will do in the systems biology section, we'll do a lot of computational analysis to see what probably the products that can be made, but we'll also be doing experimental validations of some of these things. Again, understand that this is a 1 week meeting, so it may not be possible to go into details of everything in a whole lot, but the purpose is to give you a fair idea of what needs to be done. So obviously once we build something and if it is performing the way we want, meeting all the checks and balances, then it's very good. Otherwise we need to learn from what is it not doing and then further repeat this cycle. So obviously getting a process to be made is not a simple effort but requires a substantial human effort. Now whenever we are talking about metabolic engineering, it is very analogous to actually traffic engineering, in a sense that if you want to go from and almost all of you must have used something or Google Maps or any other map based system to reach from a place to another. So if you want to get an optimal route for going from place A to place B, effectively I need two pieces of information. First of all, obviously I need a complete as possible roadmap. One can start with the basic bigger roads, but then the more information about the smaller roads we have, the better we have the option to see alternative routes. And also in order to go fast from one place to another, we need traffic information. So effectively, based on the current traffic, what's the fastest way from A to B, and this is precisely what your Google Maps does. Now this is not very different from the road maps which we use for transportation, is not very different from the road maps that we use in metabolism and biological engineering. So metabolic pathway maps are our road maps as bioengineers to design new systems and to see the capabilities of the organism. Now typically when we talk about metabolic roadmaps, they are for nowadays the most recent approaches is to make this as a genome scale roadmap. So again, this gives you information of what all the smaller pathways are also there which are present. So obviously creating a genome scale model, which is basically, it always generally starts with an organism with its annotated genome. So we need to harvest the genome or take the DNA, get it sequenced, and then with a lot of information for organism, if it is present in database, utilize this information about the annotated genome as well as database sort of information to generate what we call as a draft model. A draft model is effectively as shown in this figure. It tells you about a lot about the organism, what all is present. Based on these information, but then mostly it has lots of gaps. So certain required metabolites or products are not formed using the draft model. Many times, so again, the primary difference between a network and the model is a network is just a collection of information. A model is something that will help you simulate, that I can give it some input and it will give me some output. Now, a draft model will not do that. A draft model generally has some lot of gaps which needs gap filling. Now, this gap filling can be done with either published models, various databases, computational methods, or literature. Excuse me. So now, once we do that and we do a lot of other subsequent analysis, such as we input the experimental data, then only, as well as we conduct all these analysis through what is known as flux balance analysis, then we make what is known as a high quality model. Now, a high quality model actually has to meet a lot of checks and balances in terms of obviously a model should be able to simulate biomass formation. So if we have some information on what the organism can grow on, what products it makes, that is our first set of checks, that okay, if this organism or this model predict growth on glucose, say xylose, if the organism grows on it, and so on. And obviously what all products it makes. Does my model also simulate those? If the model does not simulate those, then we need to see what are the problems with the model that needs to be addressed. And then of course for a lot of organisms, sometimes we have information about known lethal knockouts, and this I am talking primarily about the typical organisms that are used in bioprocesses, for example E. coli, yeast, or various fungi. So we have whatever information we have in terms of lethal or otherwise knockout response. Ideally, the model that we make should be able to reproduce those responses. And of course, being from the genome sequence, it should ideally include almost all relevant and probably all pathways of interest. So for example, let's say when we talk about fungal production of citrate, then of course our primary interest is the central metabolism that produces citrate. But fungi also produce a lot of other molecules which, for example, may be acting as toxins. So we need information about that also, and if we focus entirely on the central metabolic pathways, then we miss out on those information. Can this organism produce toxins? And if we are making a bioprocess, this is a very important information to have. So of course, these are the various checks that the model goes through. In terms of balances or what we need to provide, obviously first thing is we need to have proper gap filling. Now we won't be going through the process of reconstruction of genome scale models in this course, because we will be focusing more on analysis of models. A lot of metabolic models are available through various databases and published resources. So especially for our organism of interest in bioenergy. So our focus will be more towards how do we analyze these models and less on creating those, but it is important for us to know that this will require some sort of gap filling. So for example, if this connection is not there in the annotated genome, what's the best guess we can make in terms of how these two reactions or two metabolites will be connected, and so on. So also very importantly, these genome scale metabolic models have what is also known as GPRs, or gene protein reaction information. So what gene codes for which protein, which participates in which reaction. Now just on that basis itself, this becomes very useful when you are, so it's like a full database of the possibilities of the organism. So this actually tells you which gene codes for which protein and what reaction it participates in. Also importantly, all of these reactions need to be charge and mass balanced. That means in a good model, it is essential that each of the reactions is charge balanced as well as mass balanced. So, and this becomes challenging because many times when we look into the databases, in fact, when you do the balancing of all the elements, you will realize that even within the databases there are a lot of reactions which are not balanced in mass and so on. So this actually goes through a lot of exercise in getting this correct. And then there is something in these models known as biomass reaction. Now, in a very simple terms, all this is how much of the material is required to make 1 gram of biomass. So each gram of biomass, for example, for E. coli, will have protein, lipids, DNA, RNA, and let's say some reserve sugar and so on. And this is typical for any organism. So a biomass reaction equation tells us that if I want to make 1 gram of biomass, what is the millimole requirement for each of the individual components. Now this itself is not a very trivial task, and I'll tell you why little bit later. The other information that we need to provide is what is the ATP demand or requirement of ATP for growth and maintenance. Now, as you will know that to grow itself, the organism requires a lot of ATP because it needs to make a lot of polymer molecules for growth and so on. But even if the organism is not growing, there is still a basal requirement of energy, just like we still need energy while we are sleeping. The same way, these terms are known as ATP demand for growth and maintenance. Now, in case of eukaryotes, we also need to provide the location of reactions. So again, a typical genome scale metabolic model, again a lot of which are available in published literature, have all of this information. And there are ways to check it using flux analysis and all. We'll not go there at this moment because of the paucity of time. So if we want to make a good model, we do require a lot of computational as well as experimental information. So in case of computational information, obviously we require genome sequence, its annotation, the GPR equations, charge and mass balance of reactions, and there is also another check that is required, that there should be no futile cycles. So a futile cycle is a scenario where, because it's effectively a mathematical model, there could be possibility that the model can make, because of some internal cycling, ATP without any substrate input. Now obviously that means that your whole model is wrong and you need to resolve it actually. Also, a lot of experimental data is needed. So many times there is a misconception that for making a good genome scale metabolic model, all I need is the genome information and I can make it, because there are a lot of software which are available which can do it for you. But then of course a lot of input is needed in terms of growth and substrate production, product formation rates. The biomass composition, again, the biomass composition tells you, so for this actually we need to harvest the biomass and measure how much protein is there, how much DNA is there, how much RNA is there. So each component needs to be measured, and many times this itself becomes challenging because a lot of organisms have very low amount of, say, lipids. So once if you want to, and many of them, especially the photosynthetic one, will have very slow growth rates. So to harvest, let's say, so a typical lipid percentage in a normal cell is about 7 to 8%, 7 to 10, let's keep it. Now that means if you want to, not only this, we also want subunit composition. So for protein, I want to know what is the average amino acid composition. For lipids, I want to know what's the average fatty acid composition. For DNA and RNA, I want to know what is the ATGC or AUGC content. So obviously a lot of experimental information is needed, and not just it's not entirely computational. Of course, experimentally one can determine growth and maintenance ATP measurement. A lot of studies have actually used exported for other organisms, but if we want to make a good model, we need to do this. The other thing that is generally done is substrate variety data. So there are these sort of plates which are known as biological plates, biolog plates. Now what they have is effectively, say a plate will have, single plate will have about 90-95 metabolites, substrate, sorry, and then you see what all your organisms can grow on. So that information we can go feed it back to the model to see whether again our model has all those pathways to check on. Finally, if there is knockout data available, then of course that also. So what I'm trying to say is that a good model requires a substantial human effort and curation, both experimental data as well as computational information. So they have a lot of value as such. So again, as I have told you, the biomass equation is the one that contains the metabolic requirement to synthesize all the required components of the cell. So as I have told you, we need not only the gross macromolecular composition, but also the composition of the subunits. So not only protein content, but also amino acid content. Not only total lipid content, but also lipid classes and their composition. So, and so on. Sometimes it is approximated based on nearby organisms. Now again, the good part is, at least for now, for most of the organisms that are relevant to bioenergy research, such models are available. So the focus of this course is to help you guys analyze those models and see what you can get from it. So, and again, these are some of the tools that are available for automatic reconstruction of genome scale metabolic model, and these are the references. Again, these are primarily based on what is the basic software they use. It can be web based, MATLAB based, Java based, or Python based. So based on whatever you guys are comfortable with, one can somewhat choose. But I think very importantly, one must understand that these are good for reconstructing draft metabolic models, because a lot of experimental information has not gone into it. They just make a model based on the genome sequence that we provide. So obviously, once we make these models, we'll require extensive manual curation to improve their quality. Now, why do we go about all this exercise? So the first and foremost is, using these metabolic models, one can identify unique pathways and isozymes that are present. So now this information itself is very useful, because again, during the course of metabolic engineering, there is always an interest to identify a particular isozyme which works under my condition. So many times, let's say a typical example is a process uses, or a reaction typically in most cases uses NADPH as the reducing power. But if I can identify another enzyme which, for example, does it at similar conditions but uses NADH, then suddenly this becomes better for heterologous organism compared to say the one that uses NADPH. So those are the things which are also relevant. So such information is will be there. What are the unique pathways that are present in the organism? What are the various isozymes that are present? Obviously, one can predict the growth and flux distribution under different conditions. Now, some of these you will do hands-on practice with. So we'll do some of these growth under different conditions and so on during our hands-on session. Now, very importantly, one can identify the bottlenecks in the use of a particular substrate. Now, as I mentioned, for bioenergy purposes, it's important for us to utilize both all different types of sugars as efficiently as possible. So if there is any particular lack of transporter or a key enzyme, we can simulate those. So we can simulate the addition of heterologous pathways and see whether a product can be made or not, or what further engineering is needed. So obviously, one can simulate the effect of knockout talks or expressions or introduction of. Now this is the exercise that we'll do on the third day, where we'll, in a model of E. coli, we'll introduce a heterologous pathway, and then we'll do further modeling to see what can we make a product of interest, in this case a biofuel of interest. So of course this will then identify the genetic engineering strategies to improve metabolite production. For our purposes, the metabolite is a molecule which can be used as a biofuel. Also, the genome scale metabolic model provides a lovely framework to integrate with other omics analysis. Now, I have told you a lot about genome scale metabolic models in terms of its positives, but then of course it still is just a metabolic model, and we all know that the regulation in the a cell is what actually controls a lot of metabolic flow. So any sort of regulatory information or proteomics information can be integrated with these genome scale metabolic model to further refine what we get. So briefly, I'll go with the definition of flux and what is the flux analysis does. So until now we have talked about metabolic network. Now we'll shift gears to a little bit about what is flux analysis. So if we choose a very simple straight pathway, which is from glucose to lactate, the intracellular concentration change, which is given by, let's say, d differential of the metabolite concentration with time, is given by the rate of production of this metabolite minus the rate of consumption of this metabolite in the reaction. So we can write this for each of the intracellular metabolites. Now in this case, the d-lac by dt or lactate production, which is the extracellular metabolite, versus glucose, which is the extracellular substrate. These are the extracellular fluxes that one can measure. And so what we can do is we can also set up a stoichiometric matrix which tells what is the, for each intracellular metabolite and each reaction. So 1, 2, 3, 4, 5, it has 5 reactions, so 5 columns, and row corresponds to the metabolite for which reaction the metabolite participates in, and whether it's produced in this reaction or consumed in a particular reaction. 0 means it's not participating in that reaction. So effectively, each row gives you the mass balance of the metabolite, and each column gives you the reaction stoichiometry. So this can then be written in sort of a matrix notation, where dx by dt, now in this case dx is actually any substrate and not the biomass, is the stoichiometric S here, and multiplied by the flux vector V. So under pseudo steady state assumption, which is effectively says that the rate of change of intracellular metabolite concentration is much faster than the measurement times. So this is simply reduced to mathematically S dot V becomes 0. Now this has practical usefulness, because if you take this particular example where we have 5 different fluxes, V1 to V5, where V1 and V5 are the extracellular fluxes, using the pseudo steady state hypothesis, this whole 5 set of equations can be solved by measurement of just extracellular metabolites, that is V1 and V5. So now that is the whole advantage of using the pseudo steady state hypothesis, so that we can identify the intracellular fluxes based on the extracellular measurements. Now in a real metabolic network, there can be more than 1,000 metabolite and reactions and very few extracellular measurements. So what we get is, mathematically it's an underdetermined system of equations. So this requires implementing some sort of optimization to solve it, and which is what flux balance analysis is. So FBA is primarily a framework to analyze the genome scale metabolic models. where effectively, as we have seen, the stoichiometric matrix is the way where we mathematically represent the reactions, and this is also known as structural modeling, because we do not require any sort of kinetic information here except for extracellular rates. And here what we do is we solve this set of equations by linear optimization, where we define the objective function. So what is the cellular objective, and based on that we get a flux distribution, importantly which satisfied an assumed objective criteria. So that is very important to know, that whatever we are simulating is under a certain assumption. It may not be still true, but that's what we need to evaluate. So if our assumption is the cell wants to grow at the fastest rate, then we will get a different flux distribution. If our assumption is that the cell want to minimize the total flux, then we get a different flux distribution. Which of them is true is for us to determine actually. So obviously, as I mentioned, that there is actually no best optimization criterion, but whatever solves our purpose. There have been certain studies which have looked into these aspects very carefully. So you can look at it, but then whatever optimization criteria we are trying to use, we must have a good justification of it. So obviously the flux is defined as the rate at which the substrate flows through various pathways in the cell, also known as activity of various pathways. This is very analogous to traffic flow information, which you see in Google Maps as red and green lines. Here the unit typically is millimoles per gram dry cell weight per hour, which means a gram of biomass, how much of the substrate or product it can utilize or make in each hour. Therefore, for kind of mammalian cells or other things, there can be analogous units, but we need to normalize with respect to some sort of cell amount. So obviously flux analysis is our way to see how the traffic flows, cellular traffic flows. Our group has actually reconstructed several metabolic models with various applications in bioenergy. So for example, this Geobacillus thermoglucosidaceus is a thermophilic bacterium. Thermophilic means it can ferment at higher temperature. For applications in lignocellulosic biomass to ethanol. Now similarly, a native indigenous native marine cyanobacterium, we made its genome scale metabolic model to see what is its intracellular flux distribution for making various products from CO2, and N-gaditana, which is again Nanochloropsis, is a very promising alga for making omega-3 fatty acids. Similarly, T-pseudonana, which is a diatom, so again for lipid production. Methanococcus capsulatus, which is a methanotroph for single cell production from methane and so on. So all of these, so again, what is the information what we got from it? So yes, we made all of these models, and when we analyzed it, so first of all, for example, we identified that primarily in case of Geobacillus, the model told, and which was also experimentally found, that it did not use arabinose. But what we found from these analyses is that all that is needed is a transporter. So once we added a transporter externally into the model, rest all of the pathway is available. So that directly tells us the simplest engineering that needs to be done in order to utilize the arabinose that is present in the lignocellulosic biomass. Similarly, for N-gaditana network, we could predict the formation of hydrogen. So again, production of hydrogen by these biological production of hydrogen is also of interest for bioenergy purposes. So we could predict the formation of hydrogen by N-gaditana, and then this was also validated based on literature. So whatever conditions we said that would increase the hydrogen production was in fact verified with literature evidence. And we also obviously predicted, so for many times there are alternative pathways that are for particular product, and then we predicted what is the activity of these alternative pathways. So one example that for example, for synthetic biology, what I can give you is where we reconstructed the model for the native marine cyanobacterium, and then we probed it for what is the metabolic engineering intervention that would be needed for making various products of interest. And obviously, as you will see, most of them are some sort of fuel molecule or solvents, as well as PHP is polyhydroxybutyrate, which is a biodegradable plastic. So we wanted to see, can we make this photoautotrophically, and what kind of engineering, and these are all heterologous compounds. So these are not the compounds that the organism make naturally. So in order to, for that metabolic network to make these products, what are the set of engineering that will be needed, and what is the yield in terms of mole of the product per mole of carbon can we expect. So these are the values, and then obviously these are under those conditions where the growth is not significantly impacted. In theory, one can simulate situations where we can say the growth is 5% of the maximum, and how much is the product I can get. Now, whether that's biologically relevant is an open question. So in this particular example, we have done the simulations where the biomass yield is, I think, 80% of the what is predicted under. So under those conditions, what are the yields of various products that we can expect, and what are the number of reactions that we can. So for example, if we want to make ethanol, all you need is just one reaction to be added. But then because this is one enzyme which can do two different reactions, and if you want to make something which is slightly more requires quite significant amount of metabolic engineering to make this product. So these are the theoretical yields. One can also do to further simulations to improve the yield so that no other products are formed. Now another example from ICGEB is the work done by Dr. Yassani's and Professor Fell's group, where they have done model-based improvement in alkane production in E. coli. So again, the same thing they have done. They have introduced a set of heterologous reactions. So for example, the reactions which are shown in here, acetyl-CoA to palmitoyl-CoA to pentaldehyde, pentadecane, and so on. These are the heterologous reactions. Now obviously, just introducing these set of reactions did not produce pentane, pentadecane to a significant effect. Now once they did the modeling part of it, they identified that several metabolic engineering strategies would be needed. So knockout of certain pathways, overexpression of others, and so on. So knockout of all the byproducts that are otherwise produced, as well as overproduction of the pentose phosphate pathway genes are the ones that would be needed. But then also very commendable part here is that a lot of these things were in fact experimentally implemented, which increased the alkane yield to about 94% of the theoretical maximum. So again, this is an example of what can be achievable by a good modeling exercise. With that, I hope I have given you a fair idea of what flux analysis is, flux balance analysis, with some examples. Obviously, we'll be doing a lot more in the subsequent lectures, as well as when we are doing hands-on exercises. So with that, I would obviously like to thank ICGB, Department of Science and Technology, for funding the research that we are doing, as well as BBSRC. A lot of collaborators, both national and international, I have worked with in doing a lot of analysis, and that has definitely enriched the kind of work we are doing. But of course, primary acknowledgement is to all the students who work very hard to do whatever needs to be done. Many times this requires establishing capabilities which are not generally present in the lab. So of course their hard work is greatly appreciated. So with that, thank you everyone for your attention, and I'll be happy to take any questions. So let me see what is the question. All right. So yeah, we will be, again, as I said, you guys have any question, please let me know. And this is true also for the people who are joining us online. So maybe I'll stop sharing for now, or let's see. So the question is, what is the difference between flux balance analysis and metabolic flux analysis? Now, yeah, this is a little bit of a semantic difference in the term that flux balance analysis is typically said for those computational simulations where we use optimization to solve the flux equations. So for genome scale metabolic models, we employ flux balance analysis. Metabolic flux analysis is that a scenario where we do not use optimization to get the flux distribution, but instead solve the and that is generally used for identifying or measuring the flux values in the central metabolic pathways. Most of the applications have been in the central metabolic pathways. There have been a couple of recent publications where they have used it for genome scale level, but mostly it is for identifying the activity of the central metabolic pathways, not through optimization, but by fitting least squares estimate, least squares analysis to get the estimate of intracellular flux distribution using experimental data. Now that for MFA, metabolic flux analysis, we utilize the central metabolic network and measured flux data, as well as if we are using 13C MFA, then we typically use 13C incorporation in amino acids or intracellular metabolites, depending on the kind of MFA one is doing. So there lies the primary difference. FBA optimization, MFA least square. I hope that answers it. All right. Yes. Right. 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 So yeah, so that actually becomes a little bit challenging if you are working with a newer organism, as to and even so many times what we use is we typically utilize, let's say, CAG based identifiers. So whatever we use, we need to specify what are the identifiers we are using, and generally use a single kind of identifier, and that also becomes challenging when we are utilizing the metabolic model from one set of which is based on one database, and then we have a lot of other things which are in another database. So those are the practical challenges that are there, but yeah, that is what we need to work with. So I guess if there are no further questions, I thank you everyone for your attention. We'll come back for the next talk, which will be held at 11 a.m. and so we have tea outside. So the participants are encouraged to please go ahead for the tea. Thank you. Let me send you this. Some of them some of them have physically joined in, but a lot of them are, as you can see, are joining us through Zoom. So I think we really look forward to your talk. So without further ado, I'll hand it over to you to take it from here. Thank you so much for joining.
Well, thank you so much for a very kind introduction. Now, let me see if I can get my sharing. Yeah.
Yeah, you can see.
Full screen. Can anyone confirm, please?
Yeah, we are seeing you on full screen.
Okay, perfect. Thank you. Thank you so much for the kind introduction. As you said, it is a bit early, so I hope I can deliver you some of the resources that will be useful for you during your coming course. So today I'll be talking about genome-scale metabolic model of Diatoms, and we will investigate and optimize lipid production in this organism. So I saw in the course that you all will be going through genome-scale metabolic modeling. So I thought I would introduce some basics concept of genome-scale metabolic model construction. Then we will apply flux balance analysis to investigate the lipid production in this organism. And finally, we will see how we can design the experiment based upon our model analysis. So I hope by end of this lecture you will have some fair ideas how you can relate your experiment and model and bring it together. That saves both your time and experimental cost. But before I move on to models, let's have a brief introduction to the biology of Bioenergy. So Bioenergy is a diatom, and diatoms are photoautotropic unicellular brown algae. They are a major component of marine phytoplankton and play a biochemical cycling of silica. They play a role in the cycling of silica. Approximately 40% of the total marine primary production is through diatoms, and they are responsible for 20% of global carbon fixation. So they are important from that point of aspect. However, there is another important thing why we are interested is they have a capacity to accumulate large amount of lipids in their cell, which makes them a potential a feedstock for sustainable biofuel and high-value commodities. So that was our primary interest, to get the lipids from these diatoms for biofuel or even other valuable lipids, for example, omega fatty acids. Among all diatoms, we are particularly interested in diethyl trichromatin, and the reason being, this was a project we started in 2013, and at that time, the genome sequence was available just for 2 diatoms, and one of them was diethyl trichromatin. Now, genome sequence is prerequisite for any genome-scale metabolic modeling. So that was one reason why we chose this species. Another, obviously, it can accumulate a large amount of lipids, so we were interested if we could get it at an industrial scale where we can have economically feasible extraction of these valuable fatty acids. The other important reason being is that this diatom particularly can grow in the absence of silica. Now, if there is a silica deposition on the cell wall, the experimental manipulation becomes more difficult. However, this diatom, they could grow in the absence of silica, which made the experimental manipulation and lipid extraction more simplified. And that is why we chose this species. Now, diatoms in general, they have three modes of growth. Some of you might find it very basic, but I was told that the audience is very mixed, so I will go very basic. If it is too basic, you can ignore it for now. There are phototropic, heterotropic, and mixotropic modes. So most of you might already know that in the phototropic mode, the organism converts the inorganic carbon, for example, carbon dioxide, and sunlight energy into reduced carbon via photosynthesis. This is a cheap way to produce biomass, especially even at industrial scale, because the only energy source it requires is sunlight and carbon dioxide fixation through calcium. However, this is not a preferred mode for industrial scale growth because it has many limitations. For example, it is difficult in optimizing light and gas diffusion. At a certain point, it gets limited. Then there is second mode, heterotropic mode, in which the growth is supported solely on reduced carbon. Now, many diatoms, including 5-dicarbon, do not support heterotropic growth unless they are metabolically engineered to do so. So we will not be looking into this mode in detail. However, we will be looking into mixotropic mode, the third mode, which is a combination of phototropic and heterotropic mode. It can simultaneously use light energy to fix inorganic carbon and also reduce the organic carbon for growth. The benefit is it has no requirement for optimal light penetration, and they can make use of cheap and easily available organic carbon source like glycerol. So this is highly exploited or used in the industry, so they can have a better biomass production compared to the phototropic mode. Now, apart from growth modes, the evolutionary history of diatoms are quite unique. They have different evolutionary history compared to many other photosynthetic eukaryotes such as plants and green algae. Now, they are thought to be arising from a complex intersymbiotic event. So it is believed that there was a heterotroph that then involved a phototroph, leading to a secondary intersymbiosis which gave rise to diatoms. Now, because of this unique evolutionary history, it also has unique metabolic features which are different compared to many plants and green algae. So, for example, again, some of you might be familiar with plant metabolism. If not, then bear with me. I'll go a bit more in detail so you are in the same page as others. So, Calvin cycle enzymes in plants are located in plastid. However, in diatoms, they are localized both in plastid and cytosol. The oxidative pentose phosphate pathway in diatoms is located in cytosol, and then it has two pathways, ET pathway and phosphoketolase pathway. Now, commonly these pathways are present in prokaryotes, but Subunit Composition being a eukaryote, even though they encode for these pathways. This might be difficult for some of you, so I'll go through it again, but through the network diagram, so you can capture what I was telling earlier. So this is central carbon metabolism of plants. Here is a compartment chloroplast, plastid, there is cytosol, there are enzymes, and therefore the reactions imported by these enzymes are being shown in each compartment. So R1 is Rubisco carboxylase reaction, which fixes inorganic carbon over here, carbon dioxide, into organic form. And there are a bunch of other enzymes and reactions involved with carbon cycle. There is R32 and R23. These are the reactions involving oxidative pentose phosphate pathway. Yeah, but now if we look at the central carbon metabolism of diatom, it is more distributed. as you could see, the number of reactions present in both compartments gets more evenly distributed. So diatoms, they do have a carbon cycle. R1 is present here to fix inorganic carbon. However, as you will have noticed, now R2 and R3 is no longer present in the chloroplasts. Rather, it is present in the cytosol. And then there are many more other enzymes which are shared between the phosphate pathway and the glycolysis present in cytosol as well. In addition to that, they also have phosphatidylase pathway, the one in purple over here. Sorry about this. And ET pathway, and the lower region of glycolysis present in mitochondria. So as you can see, the enzyme distributions are quite different in diatom. Therefore, for us, it was important to investigate the metabolism before we jumped into experimental design to get and optimize the lipid production in this organism. So the aim of the project was to identify and optimize potential route for lipid production. And the method that we used was we constructed, curated, and validated a genome-scale metabolic model. Since you will be looking into these models by yourself during the practical session, I will go through them with some basics. Then we applied flux balance analysis that you learned this morning to identify the potential route for lipid production. And finally, we designed our growth experiment based upon the model analysis. And the results we obtained was like we could enhance both quantity and quality of the biomass production in this organism. You can find the details in these papers. However, we will go through some of them together now. So I'm not sure how familiar you are with genome-scale metabolic model, so I'll go through it again from the very basic. So genome-scale metabolic model compared to other small or pathway models are large-size models. Usually they have hundreds to thousands reactions and metabolites. These models describe the interaction in a given organism based on the reaction network, as you can see here, predicted from the enzymes that are encoded by the genome. And there are various databases that that store these informations. For example, there is Biocyte, KEGG, and many others. So, in theory, the construction of genome scale models should be easy if we look at that aspect. You could choose your favorite organism, go to your favorite database, save or extract all the reactions from the database, and if it is your local database, this job should be done in less than a minute. So all easy and done. However, it is not easy, I'm sorry. And the reason being, any GSM or genome-scale metabolic model reconstructed solely based on database is likely to inherit a number of problems. Some of them are inconsistent naming of metabolites and reactions identifiers, incorrect reaction stoichiometries and reversibilities, and also there can be missing gene-protein reaction associations. Therefore, in order to represent a realistic representation of a given organism, an initial draft model that we will construct from a database or any other source requires refinement and curation by the user, which makes the construction of genome-scale metabolic model pipeline a much more iterative process, as you can see on the slide. So the first step is we extract reactions from the database or reconstruct our metabolic network from the aggregation. Then we need to curate or delete metabolized reactions, make corrections in either in stoichiometry of the reaction or reversibility, missing reactions. That gives us a new model, and this model should be validated for energy and mass conservation. Similarly, it should have no stoichiometric inconsistencies, and they should be able to produce biomass in the experimentally observed proportion. So if you want to build your model from scratch, you have to go through these pipelines. But there might be people who don't want to construct their own model, but rather just use the models that is available there in the literature. Still, it's a good practice that you validate your model. You check that the model can conserve mass and energy and is free from stoichiometric inconsistencies. Some of the problems that we face during the model construction from the database are that database includes non-specific metabolites. For example, they just say some tRNA or an alcohol. However, since it is a mass balance model for you, you have to make the changes or replace it with more specific metabolites. Then there can be inconsistent metabolites identifier. For example, ribose, a metabolite in the database, at least in metasite, can be present with several identifiers. For example, D-ribopyranose, D-ribopyranose. Now, if you don't check these, then you will be leading into an artificially created network discontinuity. And the results that you might get is an artifact of this discontinuity. Then there can be incorrect stoichiometry. For example, if you look at stoichiometry of reaction 3.2.1.58, there's no metabolite going into no conversion of substrate into product. So these things, again, you have to check in your model. There can be stoichiometric inconsistencies, especially this is the case in terms of polymers. So what do I mean by stoichiometric inconsistencies? Inconsistencies are if there is a metabolite A being converted to B, but if there is another reaction that converts the same metabolite A into B and C, then both the reaction or the stoichiometry of the reaction cannot be true. If C is a non-zero mass metabolite, then there is definitely a clear violation of mass conservation. So these can be checked. It is not a very difficult task to fix. There are published algorithms that you can use to find out if there are stoichiometric inconsistencies and then fix it. Similarly, you have to check if the model preserves energy conservation. Again, this is a very simple step, though essential. So again, I repeat, even if you are using a model that is already there, do validate your model before you start your analysis, because not all the published models in the literature are correct. There can be errors in the model as well. So what do we check is we check if the model can produce energy out of nowhere. And this can be done by examining the FBA solution very simply. You constrain all the transporter flux to zero, that is, there is no input flux into the system. And then you set a demand for energy production. You can do energy demand for ATP production or NADPH or NADP production or both. And if your FBA solution turns out to be feasible, means there is inconsistencies, because there is no input flux, still you are able to produce energy. And in that case, you examine all the reactions in the solution for the reversibility or any other issues, and then you can fix it manually. So these were some of the basic steps we took for the construction of genomical metabolic model of fat and connectivity. It consists of around 500 reactions and metabolites, and there are 4 compartments. Remember, Fadaikan is a eukaryote, so the model is compartmentalized as well. model. It has 4 compartments. There are specific reactions in each of the compartments. Then there is intra-compartmental transporters that exchange the metabolite between compartments. There is exchange reactions to exchange metabolites between the environment and the model. Now, in this lecture, we will be looking only at the phototropic mode. So the only source of energy for this model over here is light. And the only source of carbon is our inorganic carbon, where we simulated the model on carbon dioxide and bicarbonate. The model is, under phototropic condition, the model is able to produce all the biomass components, such as lipid, amino acid, nucleotides, in experimentally observed proportion. So this was our model, sorry. So this was our model, and after that we moved to the more fun bit, which is the analysis. For this we use flux balance analysis that you learned in the morning. Column goes quite a lot through its life cycle is light fluctuation. So they can be present at the surface of the ocean or water bodies where the light intensity is really high. However, they can also sink to the bottom of the ocean where there is a really low light intensity. So one of the earlier questions that we wanted to ask the model was, how does the metabolism changes with the light intensity in this organism? And for this, we used flux balance analysis, where our objective function was minimization of total flux. By now you would be familiar about the steady state constraint, so force that goes in. Then we added the biomass constraint. One of the difference here compared to the classic FBA is we allowed the biomass composition to vary. So the model didn't have to produce all the biomass composition in a fixed proportion, rather it was allowed to change the composition to meet its objective function. Then we added the cell maintenance cost, and also added a limit on the Rubisco reaction. So Rubisco can fix carbon dioxide, also oxygen, so it has both carboxylase and oxygenase function. However, the enzymes are saturated at some point. So the limit over here is a proxy for enzyme saturation in carbon cycle. And then we gave the photon flux, which we gradually increased to mimic the natural habitat in which the diatoms would be present. So for example, we simulated equation 1 at a certain photon flux, then we gradually increase, which you can see over here. So x-axis is the photon flux. We are gradually increasing the amount of light going into the system. Y-axis over here is the reaction flux. And as you can see, with the increase in photon or light intensity, there is increasing lipid production over here. It has certain patterns, so I have divided it into different reasons based on the flux patterns. Now, this is a mass conserved model. So if there is an output of lipid in the model, there should be input for the carbon source. So we looked at what is the carbon source for the production of this lipid. And we saw there was uptake of carbon dioxide, or increased uptake of carbon dioxide. However, this saturates at some point because there is a saturation of carbon side. Now there are two questions that arises from this plot. So there is carbon going into the system in reason B, but there is not the same amount of lipid that is being produced. So what is the fate of carbon in reason B? What happens to the carbon dioxide over here? The second question we can ask from reason C or D, that there is more amount of lipid in the carbon, then there is input of carbon dioxide. So what is the alternative source of carbon for this lipid production? So we checked other carbon transporters, which was bicarbonate. That answers our second question, that the increased demand for carbon for lipid production in region C was met by bicarbonate. And you can see it mimics the flux pattern in the lipid production. However, our question remains, what is the fate of carbon going into the system in region B? Then we look at all the exporters, that is metabolized, the model was a lab collection. And we saw that in region B, the carbon going into the system was actually being excreted as a glycolate. Now, glycolate is an end product, or one of the products, of photorespiration. So we knew there was photorespiration turning on in this system. However, it was turning off in region C, where there was demand for higher lipid. Now, again, this might be difficult to follow, so I will go through the metabolic network for each of the regions. We'll start from region B, then move to C, and then D. So what is happening in region B is there is excretion of glycolate. Carbon dioxide is being fixed by Rubisco carboxylase, but it has the dual functionality, so it also fixes oxygenase by Rubisco oxygenase. This produces glycolate. Now, higher plants can recycle glycolate. In diatoms, until recently, or until when we started, it was not very sure what is the fate of glycolate, whether it's been excreted out of the system or whether it was being recycled into the system. So in region B, we saw that there was carbon uptake, there was minimal production of lipid, that was the minimal demand. However, the rest of it was excreted in the form of glycolate. Now, if we move to region C, there is still Rubisco carboxylase reaction active fixing carbon dioxide and therefore running the carbon cycle to produce organic carbon from inorganic carbon. There is also Rubisco oxygenase reaction active. However, in this case, instead of excreting glycolate out of the system and losing carbon, it is recycling glycolate into acetate to be used for lipid production. The role of bicarbonate comes over here. It is fixed by PEP carboxylase as an impurity carbon fixation process. However, in region D, it changed. Rubisco oxygenase reaction was no longer active. Rather, all the flux was diverted to Rubisco carboxylase reaction, and this gave rise to PEP, which formed the basis for an impurity carbon fixation through PEP carboxylase, giving, again, increasing the lipid production. So overall, we found that there were multiple ways in which the light intensity was changing the metabolic process, especially the lipid production ways. So to conclude, or at least the model hypothesis for us was that lipid production can be increased with increasing light intensity and addition of bicarbonate. We did the same analysis under mixotropic condition with glycerol as organic alpha salt. So I won't go through the mixotropic again because most of them, the analysis with are the same. However, the results we found were again interesting, that even in the mixotropic condition, addition of bicarbonate and increasing the light intensity would increase the lipid production. So, based on these model results, we designed our growth experiment and we used the 2-liter bioreactor for this. So this is our experimental setup. We had two conditions. One is phototropic, where the source of energy was light, which we gradually increased from 70 to 300. There was carbon dioxide diffusion in the bioreactor. However, we also supplemented with bicarbonate. Then we had another bioreactor with mixotropic condition, where the supplementation was also glycerol. And this was all done in a fed batch culture. This is how it looked like in real life. So E10, and these are some of the results. So E10 is the media which was previously used for this organism, and the red one is the mixotropic condition, and the black one is the phototropic condition. So mixotropy, of course, increases the biomass concentration. Then we move to EE medium, which is the enhanced medium that we designed during our project. As you can see, again, the mixotropy increases and the biomass concentration. However, C is based on model analysis where we had EE medium plus bicarbonate added to it. And as you can see, it increases biomass concentration both in the phototropic but also in mixotropic, and this increase is by almost 78%, which was quite huge just by addition of bicarbonate into the system. So that makes sense because there is more carbon source, so we could not just base this on the biomass concentration, so we measured a lot of other parameters. Some of them were pain, which stands for fatty acid measurement, and EPA, which is another type of lipid. We also measure carbohydrate and the pigment present in the diatoms. And as you can see in the EE plus bicarbonate, the enhanced E10 medium plus bicarbonate, it increased both lipid and also pigments. One of the interesting things we found that it also increased carbohydrate. So this is something in future that we could engineer this strength to stop the carbohydrate production and divert this carbon further into the lipid, which could even enhance further the lipid production. We haven't tried that, but this is something could have been done in the project. Yeah, so that is all from my side. This project was part of Atp project, which was between 2013 and 16. It was a European Union funded project called Atp. All the work, or model work, was done at Oxford Brookes University, and the experimental work was done at Formental, which is an industry in France, and it was done by Bioenergy. And thank you so much for your attention. I'm happy to take any questions.
Any questions from the audience, both online as well as offline? So Dr. Dipali, I have a question. So obviously, first of all, thank you for the talk, and it was, I think for me at least, it followed very well from my previous talk, where I talked generally about the flux balance analysis and all these problems, and then where your talk followed so nicely because it talks about all these challenges in terms of futile cycles, as well as if you are using various databases, all those challenges across databases. And there was a question from one of the audience members regarding this earlier as well. So it did follow, and then ultimately with all this wonderful example of how you resolve this, and then over different photo intensities, how did you see the metabolism change as well as the product profile also change? So excellent. I think it for me it was very useful, informative, and followed very well from my talk.
Thank you.
Yeah.
Thank you. I'm glad that it made sense.
Right.
It's quite early, so I was not sure I was making any sense.
No. So yeah, in terms of my question is regarding the CO2 versus the bicarbonate question, primarily in terms of intracellular dynamics, because from my understanding is that based on the extracellular pH, which is generally implemented for phototrophic production, which is usually basic pH, where the bicarbonate is the primary species versus so what's your opinion? Is it the bicarbonate that's the primary intracellular metabolite or whatever it's a species, or versus the carbonate or carbon dioxide?
Yeah, so this is something we went through during our project. We went through this question a lot. Because we were trying the structural model, we didn't know what was the concentration of which species, whether it was carbon dioxide or bicarbonate in the system, and that was the form being used. Also, this organism encodes for enzyme that can keep the carbon dioxide and bicarbonate conversion going on. So basically, we don't know what, or we still don't know what is the dominant species, whether it's bicarbonate or carbon dioxide. However, generalized, like not going into the details, we came to know that carbon dioxide is more expensive to be used than bicarbonate, at least here in Europe. So, at an industrial scale, or when we went to Futile to do some experiment, they were happy for us to try with bicarbonate because it was a much cheaper source of carbon compared to carbon dioxide. Again, and then there comes the anaphylactic reactions that these diatoms encode for. So I think you had a very nice question, but I don't have a very simple answer because it gets more complicated. We will need more detailed kinetic model basically to answer whether it's the carbon dioxide or bicarbonate that is dominant species within the intracellular cell. Again, we didn't go through any intracellular studies, but more for us it was like make it cheaper, make it more biomass, make it quicker. So we just went with bicarbonate.
Right. And again, just during the tea break, which was just before you talked, again one of the participants asked me about this phototrophic production, why is it not viable and all, and that's where we were discussing that. Of course, it's almost impossible to make biofuel molecules cost effectively under phototrophic mode. So most of the product is in terms of high value lipids. Right. Actually, again, I think, is there any other question from the audience? Because otherwise I'll ask one more. Yes, great. So there is one more question from the audience. No,
Hello. Thank you, ma'am. That was a nice talk. So my question is regarding the validation of the genome scale model. So you said that your model was able to predict at some conditions. My question is, when do you say that your model is fit and it's ready to make prediction? Let's say you have also a good MIMOT score, but I have only one condition at which it predicts. Could I say that my model is fit for making predictions? So how do you draw the conclusion by saying that my model is now validated and it's fit for making predictions or simulations?
Okay, cool. So we follow, or I personally follow, two-step validation. One is theoretical validation, where you just look at the theoretical bit of your model. And these are pretty straightforward. You can use the You can write your simple script or even use, in some case, FBA to use it. So the things that we check is all the reactions in the model is mass balance. So for example, there is 5 carbon, 5 nitrogen on your left-hand side. There should be 5 carbon, 5 nitrogen on your right-hand side. Basically, your reaction has to be balanced on both sides. Check it for every reaction, because database, they were not meant to be used for model construction. So they don't handle many reactions in terms of mass conservation very well, especially if it is polymers. I did show some of the examples that we have in polymers, but in the database you can find that there is 5 units of glucose going into 10 units of starch or something. So there is definitely a violation of mass conservation. So you can check that, that's the theoretical bit. Then as I mentioned earlier, check for the mass and energy conservation. So your models should not be able to produce mass out of nowhere. Just block all the input to your system and see if you can still produce the mass balance component. Similarly, if you can still produce the energy. And if you can, then you have to look into the solution to see what is the cause of that issue, whether it's the reversibility, whether it's the stoichiometry, and you can go and curate those things. Then there's already algorithm that will check whether there is any stoichiometric inconsistencies again. So those were the theoretical bit. Once I have my theoretically validated model, then I am more confident that model at least theoretically is good enough. Then of course we are trying to investigate the phenotype from the models. And the phenotypes can vary, it's more complicated. There are too many parameters in the real life that can affect the phenotypic behavior. So at the second stage, I look at the experimental data. So for now at Portland Institute, I have access to a lot of experimental data, and I fully utilize that. And I would suggest the same to use that if you have access to any experimental data, just use it in the model. It doesn't have to be for your project, just for the validation or checking whether the model behaves similar to what you would expect. They might not behave, of course, because this is a structural model, and most genome scale metabolic models are structural. We don't have kinetic information for all ranges. But at least you know why it is behaving the way it is behaving. That gives me confidence that, okay, I'm not using model in a wrong sense. I hope I answered the question.
Okay. Thank you, ma'am.
Hello. Hello, ma'am. So as you mentioned that as the light intensity increases, the lipid production also increases. So is there any optimal range for the photon intensity, or is it like if we keep on increasing the photon intensity, the lipid production will also increase?
I'm sorry, like your voice was really breaking for me.
Hello.
So is there someone who can repeat it in a clear way? I understood it was related to photon intensity, but I didn't get the actual question.
Yeah, so I guess that the question was whether there would be a monotonic increase in the lipid production with light intensity, or would you get a sort of a saturation? What is the expectation of response to light intensity of lipid accumulation?
So in terms of model, you would get a linear increase because we are doing linear programming. So as you would know, flux balance analysis is based on the linear programming. So unless you impose a constraint, you will just see a linear increase in the model. However, that is not the case in the cells, because there are many factors that limit this. For example, there will be the cell capacity or the volume of the cell, there will be enzyme saturation, and many other factors. So though in the model you will see a linear increase, it will not be a linear increase in the real life, or at least I don't expect that.
Good morning, as you mentioned that increasing carbonate as the carbon source really helped in increasing the biomass, the lipid content. So my question is, did this have any adverse effect on like the cell growth because of pH toxicity, or was there an optimal concentration of carbonate that was maintained?
So the concentration that we use didn't have any adverse effect, or at least we didn't see because we measure different parameters, not only biomass and growth, but also. But yes, over concentration could have a negative effect, though we didn't see at our concentration. It goes the same with the light. We didn't see any damage at our light intensity, but of course that light intensity could go more higher, and we do know if that would cause photo damage if we would have increased further. In the literature, you can see that the light intensity of like 1000 or more than that has been used. We only went until 300, if I remember correctly. One of the reasons was we didn't have the light panel more than that. Though we wanted to go higher than that to see if we can provide more light intensity. So yes, in both cases, light and bicarbonate, or even other metabolites, for example, nitrate, sulfate, phosphate, other elements, I think you have to optimize it. At a certain point, it might reach a concentration where that will do more adverse effect rather than only good thing.
Right, thank you, ma'am.
Hello, ma'am. Next question is mine. Ma'am, as you said, photon and bicarbonate ion is mainly responsible for the higher lipid production and biomass. But my question is that have you ever studied that carbon dioxide means increasing concentration of carbon dioxide during fermentation and bicarbonate ion both have synergistic effect on cell, or what you get during a fermentation?
So we did check increasing the carbon dioxide instead of bicarbonate. So giving the same amount of carbon, it would just inform of carbon dioxide. We didn't check in our project, though this is something I wanted to check to see if it was the bicarbonate that is making the difference, or it could be any form of carbon, inorganic carbon. One of the reasons was that our experimentalists, they told us that carbon dioxide was much more expensive than bicarbonate, so we should stick with bicarbonate. Though there are some studies in the literature where they have increased carbon dioxide without bicarbonate, so they are still increasing the inorganic carbon content, but just solely by carbon dioxide. Though they didn't see the same amount or similar proportional increase. So we don't know yet whether we could have reached the same biomass and lipid solely based on carbon dioxide, or whether it's the bicarbonate, because there is an aperitif enzymes that has better effect on the biomass lipid. This is something that will need more detailed experimental validation, which we haven't done so far.
Okay, thank you.
Are there any questions from our online audience? There is one more from our offline audience.
Good morning, ma'am. Ma'am, my question to you is that the best conditions that you have described, under those conditions, what is the highest amount of lipid that you could get in terms of yield?
Sorry, I'm not able to get the question.
So the question is, under the optimal condition, what is the lipid yield you got? So maybe carbon to carbon or gram to gram basis.
Yeah, I'm not sure if the results I showed you are in use, but you can find it in the paper, definitely, because we looked at yields. Let me check. So currently I have the values in milligram per liter per day, and that was like way higher even in terms of lipid. I think it was much higher when we added bicarbonate compared to without bicarbonate. But yeah, for further values, I'm sorry, I have to go through the paper again. It was a bit a long time ago, but I could get back to you if that helps.
That's fine. So again, any question from the online audience, if you guys want to ask, this would be a good time. That's it. I think this is a very relevant question in the text box.
So it depends. So our genome scale model, at least the first draft, depends upon how good your genome annotation is. So it depends, and especially if it is a non-conventional organism other than E. coli, most of the annotations are not very well. There are a lot of enzymes which are not annotated. So yes, it is challenging. Though it depends what is the research question you are asking to your model, that's gonna make it more challenging for the model curation or update. So in this case, the annotation was fairly okay, so it was not a challenge as much. However, it depends, and also we were looking mainly on the primary metabolism and lipid, which is better than if you are interested in secondary metabolites, which are way less well annotated. So if anyone is looking into pigments, that can be more challenging because many of the pathways will not be annotated. So yeah, I mean, this annotation software nowadays are pretty good. No, I won't say the best. However, it still needs the manual curation and annotation, and same is with the genome scale metabolic models because they are derived from the annotation that we get from the software. So, yeah.
Yeah, please go ahead.
You could unmute yourself. I'm not sure.
Okay, thank you. I was wondering about the curation time of your model.
Sorry, what time?
No, the curation time, regarding the curation time of the model.
Curation time.
Curation time, sorry. Yes, so we had two versions of the model. One was the published model from Roscalson from Rotterdam State University. So when I started this research work, I started with this model. So there was already some model for me, though I updated the model for more new reactions and updated annotation. Then we also built a new model from the scratch, from the database, just to see how much work and difference there was. So it depends upon the experience level. For me, it might have taken a month. But if I do the same with the different organisms, nowadays it takes me a week. And if the annotation is good, it might take just a few days. So it depends upon the experience. Since that was my first model, it took me around a month or more, I must say. So it is time consuming, especially not the construction, especially the curation bit, because once you know that there is some inconsistencies, you have to investigate each of the reactions that causes the inconsistencies and have to curate them. So that was more time consuming compared to the construction.
Thank you,
Does
Carlos,
that
for the
answer
question.
your question, Carlos?
Yes, thank you.
No worries.
Are there any other questions? questions? I guess in the interest of time, we may want to move on, and then definitely thank you, Dr. Singh, for this very wonderful and very relevant talk for the workshop course, and thank you for taking time this early in the morning, and thank you again.
Well, thank you so much for having me, and good luck with the course.
Thank you. You can start here. So yeah, for the online participants, and as we have emailed, unfortunately it will not be possible to do the afternoon sessions online, just because a lot of them require experimental sessions in the laboratory. We are evaluating the possibility of having some things recorded and then shared at a later date. So again, we will meet tomorrow morning at 9:30 for another set of very interesting and exciting talks. So please log back in tomorrow morning at 9:30 a.m. Before 9:30 a.m. IST for the next talks. Thank you. The one more announcement is today around 4 a.m. or so we'll have our special introduction by the director of ICGB. The final time we'll let you guys know. So in case you guys want to, any one of you want to join over that would be wonderful. Thank you. Please do not log out for a minute, please. I need to check who all are present. All right, thank you everyone. You may log out now. Yeah. See you tomorrow.